Okay, let's look at chapter 21. So this goes with chapter 19 in that we're looking at development and how to turn on genes on and off in development. So for this, there are 3 different steps in development. The first one is cell division, so we simply become more cells big. The second one is cell differentiation. And that is where the cells become specialized, and so certain genes are turned on and certain genes are turned off, and that's what allows cells to become different. And then lastly is morphogenesis. And with this, that is when we start to form shape, all right? So they start to take form. And so the cells aren't only different, but they start to form differently. All right. So let's look at these things called morphogenic events. And so remember that plants and animals go through morphogenesis and cell division differentiation at different points in their life. And so if you recall from our notes, this particular picture here where it looks at cell division, morphogenesis, and cell differentiation can all occur together at the same time. Usually cell division starts out first, and then then we start to get into differentiation and so on. But eventually all 3 are going on simultaneously, going from zygote to adult. All right, so we want to review that. All right, let's look at specifically some more in more detail about cell differentiation. So cell differentiation, remember that there is things within the egg cell, within the egg cell. So remember that those are cytoplasmic determinants. And these cytoplasmic determinants, depending upon what type you have, turns on and off certain genes eventually in the cell. And so that was this picture here. So remember, here's your egg cell, has a different collection of cytoplasmic determinants, and they're not evenly spaced. And so what happens when the sperm fertilizes the egg, the zygote begins to divide and divide by mitosis to grow. And each new cell, because the determinants weren't evenly spread throughout the cell, this cell has different chemicals and different cytoplasmic determinants than these. And so therefore these cells will develop differently because these determinants turn on different genes and different sets of genes. So that's your cytoplasmic determinants, and so review that. And then remember things in the environment around the zygote. So different molecules like hormones, or in this case here, this is what the second part of the picture is that cells adjacent to one another can release molecules that communicate with other cells around it. So this cell is communicating with this cell by releasing molecules, and then they bind to receptors, which turns on certain genes in the nucleus. So the environment, molecules in the environment bind to receptors and turn on different genes. So they bind to certain receptors, and so both of these turn on certain genes depending upon the molecule and the gene and so on and so forth. All right, and so that's basically the egg cell. Then we talked about something called determination. And so determination is the cell is irreversibly committed to becoming a cell type. That was this picture here that we talked about here. So once certain genes are turned on, so an environment turns on control genes. Once certain genes are turned on, the transcription factors activate other genes, and once those genes are on, that cell will become a muscle cell instead of other cells. So remember, it can start out as an embryonic precursor cell, and then once certain genes are turned on, then it becomes irreversibly committed because the transcription factors are made. And what do those transcription factors do? They turn on the genes, in this case, to activate genes for muscle proteins, and so therefore that cell will become a muscle cell. So there is a point in time where you can't go back. All right, and so that was determination. All right, then we looked at pattern formation. So you want to look at pattern formation and, you know, how the head becomes the head and the tail becomes the tail, and so on. And so remember they looked at and they studied fruit flies, and so looking at patterns like head, thorax, and abdomen. And I showed you that picture of what happens when you have a mutation that they study. Flies with, you know, the legs coming out of their head instead of the antenna, and so they look at mutations and see what has happened, and that's how we learn about genes. And so one example of a maternal effect gene that encodes for cytoplasmic determinants is the bicoid gene. So so the cytoplasmic determinants, an example of that, all right, is the bicoid gene. So the bicoid gene codes for making the bicoid protein. Codes for bicoid protein. And so remember that this picture here, that the collection of the bicoid protein, this is the egg cell, and then the egg becomes fertilized, so here's fertilization, and you have the bicoid protein in one side of the egg cell, and then not in the other side, so this side will become the head, this will become the tail, and so it's where these proteins are. All right, and so that also helps with axis in determining head and tail. And then we looked at apoptosis. And remember, that's a review of the deliberate killing of cells, like between the fingers and so on. And then the last thing we looked at was the homeotic genes. And so the homeotic genes, we did that at coloring book activity. So homeotic genes. And homeotic genes, this is with we looked at the fly versus the mouse and how they have the same type of genes that are turned on and encode for different parts of the body. So look at your color, this activity here that we did here with the color coding and so on, that they have the same color coded genes that code for similar spots on the fly versus the mouse, and so those are conserved throughout time. So you have very few genes that code for the spacing of the various spots of the organism, and that those genes are the same across lots of different organisms, like this one from a fruit fly to a mouse. Notice the mouse has duplications of that, but it's the same genes, and so across with that. And so review those particular genes, and remember we wrote this as well when we're talking about homeotic genes, that homeotic genes have a regulatory protein, and a homeobox is part of that, so that when you transcribe and translate that protein molecule, that homeobox codes for a certain section of the molecule called the homeodomain that helps initiate transcription and acts as a transcription factor there. And so that's another way to control development and turning on and off genes in the cell, and that's how the homeotic gene turns on genes, and so certain genes are turned on, and therefore it will develop into a specific type of cell. All right, and so that's basically chapter 21 in a nutshell. So you just have to look through, you can read through chapter 21, the sections of the book that talks about this, and so on. So that's a quick little review. All right, I'll see you later.